So we have the astrocytes. These are star-shaped cells. Their processes are often in contact with the blood vessels through pure vascular photoprocesses, and they form part of the blood-brain barrier that separate the neuronal tissue from the blood. Astrocytes provide mechanical and metabolic support for the neurons in the central nervous system and involved in the removal of transmitter from the synapses and metabolism of these transmitter, and responsible for scar forming in the CNS. As we mentioned earlier, such as injury to the nervous tissue is replaced by scar tissue produced by the astrocyte. Astrocytes in this figure, they are continue profuse cytoplasmic processes that in close contact to the blood vessels, forming what's called the blood-brain barrier. In this figure, we can see different part of the astrocyte with their cytoplasmic processes and enclosed to a blood vessel. This is section in the cerebral cortex that is stained by Golgi stain, which is specific for neuronal tissue. In this figure, we have immunohistochemistry staining for glial fibrillary acidic protein of the astrocyte. This is the glial fibrillary acidic protein appear in the active astrocyte that activated during the process of scar formation, not in quiescent astrocyte. So they appear in the cytoplasmic processes and it's involved in the scar formation in the injury of the cerebral cortex. So we have astrocyte of two types, subtypes. One is called the protoplasmic astrocyte that containing many branches which are short in length, and they are found in the gray matter of the CNS, and they have intimate contact with the blood vessels. And we have a fibrous astrocyte in which the branches are and longer, and they are less in number and chiefly found in the white matter of the nervous system. So the protoplasmic astrocyte profuse branching with short in length, and the fibrous astrocyte, they are longer branches and less in number. Both have a contact with the blood vessels, forming a blood-brain barrier. Oligodendrocyte, they have smaller than astrocyte and have fewer and shorter processes. They are responsible for formation myelin sheath around the axon in the central nervous system, and it is synonym to the Schwann cells in the peripheral nervous system that forming a myelin sheath. The oligodendrocyte forming Schwann cells, forming myelin sheaths surrounding several axon segments at the same time. So the white matter, they form myelin sheath around many axons. In the gray matter, may lightly myelinated some dendrites. They have intimate association with the neurons, and so when they are cultured with the neurons, they are responsible for growing in the neuronal tissue within culture. So in this we have the cell body. We have different processes of oligodendrocyte that forming myelin sheath around different segments of neurons, and this is another figure showing the cell body and their processes that forming a myelin sheath or warp surrounding the axon or nerve fibers. The Schwann cells are a synonym which is responsible for myelin sheath formation around the axon segment, and it is found in the peripheral nervous system. The myelin sheath produced by the Schwann cells is not continuous, it is discontinuous or segmented at a region called a node of Ranvier. In this section, we can see a nerve fiber section in the nerve bundles containing different nerve fibers. We have a myelinated nerve fiber that's surrounded by myelin sheath that appear light in color, and in the center we have the nerve fiber or exoplasm. Here have a myelinated nerve fiber. Unmyelinated nerve fiber is devoid of myelin sheath and it's only covered by a layer called the endoneurium. In myelinated nerve fiber, we can see the nucleus of the Schwann cell. So this is the nerve fiber or exoplasm, this is the myelin sheath, and this is the nucleus and the cytoplasm of Schwann cell. So it is in close contact to the nerve fiber. So the white layer found in myelinated nerve fiber, and the unmyelinated nerve fiber only surrounded by the endoneurium, and the nucleus of Schwann cells found in close contact to the nerve fiber. In the EM, we can see by a specific stain called osmium tetroxide, we have the lipid material stained in dark black color. So the myelinated nerve fiber is surrounded by a black layer, which is the myelin sheath found in the myelinated nerve fiber. Ependymal cells, they are responsible for lining the cavities of the central nervous system, this including the ventricle of the brain and central canal of the spinal cord. They are found in form of single epithelium ranged from cuboidal to columnar in height and contain cilia in their frail luminal surface. And they are responsible for the circulation of the CSF, which is the cerebral spinal fluid. In between, they lack tight junction between ependymal cells that allow free exchange between the cerebrospinal fluid and the nervous tissue. So we have a single layer of cells in form of cuboidal to columnar cells and the free ciliary processes. Here we have a single layer of cells. We have the cilia that project to the luminal surface. Microglia, they are part from the mononuclear phagocyte system. They are of mesodermal origin, while other cells are of ectodermal origin. They are the macrophage precursor that circulate in the bloodstream. They are responsible for phagocytosis of foreign different particle or form damaged tissue. They are differ from other glial cells by formation of by found of their elongated nucleus, while other glial cells have a spherical rounded nucleus. Here we have a figure of microglia. We have elliptical elongated nucleus and profuse cytoplasmic processes that found close to the neurons. Synapses, they are specialized contact between neurons in the neuronal circuit, and they are responsible for unidirectional flow of nerve impulse. The synapse, in which we have presynaptic area and postsynaptic area that's separated by synaptic cleft. Types of synapses include chemical synapse, in which we have neurotransmitters, and we have electrical synapse, in which we have a gap junction that's responsible for electrical coupling. Synapse, we have the synaptic vesicles that containing the neurotransmitter, which found in the presynaptic area, and the synaptic cleft, which found between the pre and postsynaptic area, and the synaptic and the postsynaptic area we have receptors for the neurotransmitter. In this figure, this is a figure of chemical synapse that demonstrate the presynaptic area, which is containing a vesicle studded with a neurotransmitter, and we have the postsynaptic area, we have receptors for the neurotransmitter. So when action potential reach to the nerve terminal, there will be change in the action potential. There will be exocytosis of a neurotransmitter toward the synaptic cleft, and this is the neurotransmitter will be bind to the receptor on postsynaptic membrane. Sometimes we have there is an enzyme that is responsible for clearance of this neurotransmitters from their receptors, such as in the clinical condition called the myasthenia gravis, in which we have the neurotransmitter acetylcholine, and we have enzyme called cholinesterase responsible for clearance of receptors in the postsynaptic membrane. Deficiency in the cholinesterase lead to repetitive stimulation by the same stimulus and the progressive weakening of skeletal muscle. Synapses found in different forms, such as we have axon with dendrite called exodendritic, or we have axon with a spine of dendrite called exospinic. Each with all of them share the same principle, which is a presynaptic membrane filled with vesicles, and we have synaptic cleft. Also, they have a common structure. So types of synapses include exosomatic, exodendritic, and exoaxonic. This demonstrated in this figure. So we have exosomatic, we have axon with the nerve cell body or pericardium, or with the dendrites, such as with dendritic spines, such as exodendritic or exospinous, or we have exoaxonic between adjacent axons. All of them share the same principles, such as we have a presynaptic area studded with the vesicles with the neurotransmitter, and we have high contact of mitochondria since the process is energy dependent, and the postsynaptic area containing receptors for the neurotransmitters. And thank you for your listening.